Welcome to APS seminar. We'll get started for today. My name is Jim Anderson. I'm faculty in the Department of Agronomy and Plant Genetics. And our speaker today, Prabin Bajgain, works under my supervision. So I have the pleasure of introducing him today. So as is typical for our APS webinars, we like to hold questions to the end. You can type your questions into the Q&A box, and then I'll moderate them at the end of the presentation. Or if you raise your hand at the end, we can open up your mic and you can ask your question live as well. So Prabin is a native of Nepal. He did his bachelor's and master's degrees at Brigham Young University. He came to us in 2011 to start a PhD program that he finished in 2014. He did stem rust mapping of wheat, and he was the first to use next-gen sequencing on our wheat breeding and genetics project. And he was also the winner of the 2015 Hayes Phillips Award. And after he graduated, he was a hybrid rye breeder with KWS in Germany in 2015, came back to the US and was a postdoc in the Department of Agronomy at Purdue University with Mohsen Mohammadi from about December 2015 to June of 2017. And then he came back to us as a postdoc in June of 2017 and has been serving as a Kernza breeder and been doing research predominantly on intermediate wheatgrass or Kernza, but also doing some wheat work as well. Part of his funding has come from wheat research in the last few years as well. He's had 6 senior authored publications since coming back to us in 2017, so a high level of productivity. And he's been very good at writing grants as well. He was the primary author of a $2 million FFAR grant that was awarded a year ago, a key contributor to a $10 million Kernza Cat grant led by Jake Youngers in our department. And he was also the key author on Forever Green Initiative, Minnesota Department of Agriculture, and General Mills Foundation grants as well. So Prabin is going to restrict his talk today just to intermediate wheatgrass because he's got more than enough material there. So we decided to leave wheat out of this. So Prabin, I will turn it over to you.
All right. Thank you, Dr. Anderson, for the introduction. I'd also like to thank Dr. Donwise for this opportunity to present. And I think this may be the first time wheatgrass has presented on an overall breeding level in the department, so quite exciting. So the title of my talk is Genomics Assisted Breeding for Trait Improvement in Intermediate Wheatgrass. In parentheses, you can see Kernza. Kernza is a trademark name for approved intermediate wheatgrass varieties, primarily for food usage, and the trademark is owned by the Land Institute. However, for the context of my presentation, I will probably say Kernza, intermediate wheatgrass, or wheatgrass interchangeably. So for the context of this talk, they're all the same, but I'd like to know that that means approved varieties of wheatgrass, mainly for food consumption. And I focus most of the talk in the work that I have done over the course of, I guess, almost 4 years now. But I'd also like to highlight some additional work that has been done by the wheatgrass community as a whole, just to share some progress we've made in terms of tool development, their adoption, and usage in breeding programs. So this is the rough outline for the talk today. I'd introduce the species to those who are not too familiar with it. Then we'll go on and discuss wheatgrass domestication, some resource development in the crop, and then we'll dive into some breeding topics, and I'll focus that part of the talk to discuss genomic selection or genomic prediction for trait improvement, release of MN-Clearwater, which was our first Kernza variety. And then I'll discuss 2 projects that have utilized these genetic and genomic resources. The first will be a kernel hardness project, and the second would be disease resistance project. So what exactly is intermediate wheatgrass? The species name is Thinopyrum intermedium. It is a perennial relative of wheat And it has a high biomass and extensive root structure, as you can see in this picture in the middle here, courtesy of the Land Institute. On the left, we have winter wheat, I believe. And just by comparison, you can see how deep, dense, and just extensive that root mass is. It has impressive winter hardiness. If you did not know, as we breed it here, we have been doing so for the past 10 years. There's a breeding program up in Manitoba, I believe, and between the two programs, we've seen little to no winter kill. So pretty good for the environment and the climate we have up here. It has been used to introgress some disease resistance genes to wheat. Some stem rust genes come to mind. I think BYDV is also from wheatgrass. Like wheat, it is an allohexaploid with 21 chromosomes, and the estimated genome size is 13 gigabase pairs, slightly under wheat, which is about 16. And just like wheat, it has 3 progenitor species. The diploid Pseudoregneria spicata, shown up here, is theorized to have intermated with a segmental tetraploid from these two, which are Thinopyrum basarabicum in the middle and Thinopyrum elongatum at the bottom here. And the hybridization has given rise to our modern day wheatgrass crop. It is a near obligate outcrosser. You can self it, but the plant will exhibit severe inbreeding depression. So selfing is pretty rare out in the wild, especially when there's a big pollen pool present. And by the virtue of being an outcrossing plant, many loci are heterozygous. In my own populations, I've seen heterozygosity ranging from about 35 all the way to 65%, depending on the population, the loci that were sampled during genotyping and such. And in theory, again, because of the outcrossing nature, each seed is heterogeneous. So each seed is, in theory, different than the next in the lot. So why do we care about this crop and why are we trying to domesticate it? There's a number of reasons, and I'll start with the ecological benefits. As you might have heard, it offers a number of them. So first of all, let's look at the root structure again throughout the year. Here, although it says September, this is not a first-year plant from my experience, probably second, if not third. But compared with a winter wheat, where in September it's almost non-existent, if you move along, wheatgrass has that amazing root presence under the soil, whereas wheat or rye, even for that matter, not so much in comparison. And because of this extensive root mass, wheatgrass is able to reduce the amount of soil erosion, nutrient runoff compared to its annual counterparts, and it also has improved carbon sequestration and improved microbial activity. Basically, a better improved soil health that's offered by this plant. As humans, we care about food, obviously. Wheatgrass flour and seed have been used to make a diverse array of food products. Up left here is Birchwood Cafe, a local restaurant, I believe some pancakes, but they also make really nice dessert bars. I'm not affiliated with them, but if you have time, definitely go check it out. They make really good products with Kernza flour. There's also Kernza bread, chips, noodles, cereals, and so on. And some of these are local, some are national, so both local and national interest in terms of using its flour to make food. Another food type that we probably care more about is beer. And we have Bang Brewing here, just a couple miles south, close to Energy Park. They make amazing Kernza beer. And it's not 100% Kernza flour, it's in a mixture, I think, pretty sure. Keep the North Cold was a beer made by Fair State Co-op, got discontinued. Patagonia, I'm sure you've heard the name, they made, and I believe still make Long Root Ale, also pretty good beer. And then there's other beer, like there's not everything I could fit in this slide, but there's a number of initiatives, local and national again, that utilize the Kernza flour to brew beer. And they all, the ones I've tasted, they all taste pretty good. For our animal friends, they also provide excellent protein content, either through grazing or as hay. I think the protein quality compares that with the standard hay materials. So from ecological benefits to human to feed, wheatgrass has a lot to offer. And for these all reasons, we're working on it currently to make it better. A brief look at the timeline of its domestication. It started as a forage crop in North America in early 1900s. The Rodale Institute in Pennsylvania in mid to late 1980s selected intermediate wheatgrass out of about 100 different perennial species as a prime candidate for domestication, and they did so by evaluating its agronomic traits, such as seed size, height, resistance to lodging, and also the potential food usage. They evaluated, I think, about 250 accessions, and here it underwent 2 cycles of selection, after which the materials were inherited by the Land Institute in Kansas. And since 2002, they've maintained an excellent breeding program that's led by Dr. Lee DeHaan. And we got our materials in 2011 from the Land Institute, about 2,500 seedlings from 66 mother plants. Since then, we've completed 4 breeding cycles. Each cycle is about 2 years for us. We planted our 5th cycle last fall, so we'll be harvesting this summer for the first time. But the highlight of our work in the past decade probably has to be the release of MN-Clearwater, which was our first Kernza perennial grain cultivar. This was released in summer of 2019, and since then has gone on to be planted in hundreds of acres. I know, which is probably nothing compared to other mainstream crops, but the adoption has been quite phenomenal within Minnesota, outside Minnesota, and as a matter of fact, outside the United States. So quite exciting time to be working with this crop. Now, if you look at the timeline, you can see that the first selection was done in the 80s, and 2020, we're only 40 years from the first selection. However, the intensive process did not start until 2002, when the Land Institute started selecting and breeding this crop. So effectively, only about 20 years of good, rigorous breeding. And compare that with something like wheat, which has seen, I think my slide froze, there we go, which started domestication process thousands of years ago. However, despite being a new crop, our targets haven't really changed. We want short plant height, good spike density, good seed size, reduced shattering in the field, but free threshing out of the combine and such. The difference, as I mentioned, is that wheat had thousands of years to morph into the crop that it is today, whereas wheatgrass, we're on a much shorter timeline, as we want the benefits to be available to the growers, consumers, and regular folks like us ASAP. And that would be the focus of my talk today, to show what tools we've developed that will help us try to achieve this goal. The figure on the right is a comparison of seed size of modern wheat on top row and the progenitor seeds on the bottom. You can see modern wheat is pretty plump and big compared to the progenitor seeds, pretty skinny and long. Wheatgrass seeds currently do not look very different from what you see in the bottom row, and I'll show an example slide later. And this gives me hope that, with all the tools that we are developing, we can try to catch up. Probably not in the next decade or two, but hopefully it doesn't take too long to get to this phase. Now, before I jump into breeding and traits, I kind of want to take a step back and describe some of the advancements that have been made with regard to wheatgrass genomics and genetic tool development, and share how the wheatgrass community since nearly 2015, 2016 has been rapidly using these tools to improve the crop. Now, I picked this study because it's a good starting point as a reference to compare wheatgrass research that was done up to about 2015 and the research that was done after 2015. Here you can see that they use SSR markers to study wheatgrass's genome evolution. Now, that's not because next-gen technologies weren't available. They identified these SSR markers based on cDNA sequence using high-throughput machines. And I'm also not saying SSR markers aren't good. For the purpose of this study, I think they serve these guys pretty well. But I mainly want to sort of compare how fast and how readily the wheatgrass community developed genomic and genetic tools and adopted them to improve the crop. And as a first example, we will look at the year 2016. This study came out of our own group done by the previous Kernza breeder. And what he did was, for the first time, optimize genomic selection models to expedite the domestication process of wheatgrass. He used about 1,100 genotypes from the first cycle at the University of Minnesota, and evaluated them in 2 years in St. Paul only. The traits he looked at were yield, seed size, free threshing height, and biomass. I think there were others too, but these are the ones that were focused on this study. And the results basically recommended that in order to get good predictions in wheatgrass, you need about 30 to 40 families with 6 to 8 individuals per family that have been genotyped over about 2,000 to 3,000 markers. I'll go over what genomic selection is in a few slides, but I'd like to tell you that we have pretty much been following this recommendation, except that we just bumped some numbers up. So instead of 30 to 40 families, we use about 70 families currently with 8 to 9 individuals. And as for the number of markers, it varies from one cycle to the next, but on average, it's probably between 7 to 10,000, sometimes goes as high as 13 or 14. So a year later, in 2017, the wheatgrass community developed its first consensus map. A number of collaborators, Kansas State University, the Land Institute, University of Minnesota, and USDA ARS in Utah. The figure here shows alignment of wheatgrass linkage maps with that in barley chromosomes, and overall pretty good quality. I think the populations used were developed at the Land Institute, Minnesota, and USDA. And because of the good quality of the map that was developed, it has been used in a lot of QTL mapping studies since then, and was also used to anchor reads during the genome assembly, which takes me to this slide. We have a chromosome level assembly genome reference available for wheatgrass. It's currently draft version 2.1. It's open access to all wheatgrass researchers for mapping or marker discovery, other genomic studies. And this work was led by Dr. Jesse Poland at Kansas State University and his then postdoc, Dr. Kevin Dorn, and collaborators from the Land Institute, Dr. Lidi Han and Hudson Alpha. I have been told that the next version is under development and has much larger N50 values. So the quality of markers we will be identifying in the future should improve quite drastically, actually. So with the tools that have been developed, both the consensus map and the genome sequence, there's more than 500 QTL that have been discovered in wheatgrass for a number of traits ranging from height, heading to yield component traits and domestication traits. I'm showing only 2 examples here. This is from the University of Minnesota, and this is from USDA Utah. But there's other studies as well that have reported a lot of QTLs. Another, excuse me, there's some technical problems. My slides are stuck. Oh, that looks like I can see. There you go. Another use of the reference genome is to identify candidate genes, because the genome 2.1 has been annotated. It's just a short table here with some examples. There's brittle rachis from barley, shattering from rice, reduced height from wheat. And I'm sure this is not an extensive list, but you get the idea that because of the genome sequence that's available, we can identify genes that are homologous to annual grass crops and genes that are important in either domestication or other agronomic traits. The most use or the widest use of the tools discovered so far probably has to be genomic selection. However, I'm sure a lot of you are well versed because a lot of faculty and students and postdocs do work in genomic selection, but allow me to go through it one more time. There is a training population, which is a smaller than your overall breeding population, but resembles very closely the breeding population. That is phenotyped for traits of interest in set number of locations, years, reps. And the phenotypic data is shown by Y. It is genotyped using genome-wide markers shown by X, and you regress the data on markers to estimate marker effect shown by beta. The breeding population is genotyped only, not phenotyped, for the same set of markers. And this formula is reversed down here, where once you have the markers, you apply the marker effects to get Y, which is the predicted phenotypic values. And once you have the predicted phenotypic values for the entire breeding population, you can select the best ones to either plant in the field or cross in the greenhouse. It depends on the scheme you have. It is, I would say it still is a fairly new technology in plant breeding, and it shows the promise of improving breeding efficiency at a rate not possible probably 10, 12 years ago. And especially for a perennial species like wheatgrass, where our life cycle is 1 year, we plant in September or late August, and we don't harvest until early August. It has been a blessing. It saves a lot of time and a lot of energy inputs. One example use of genomic prediction, genomic selection, was across 2 breeding programs, Minnesota and the Land Institute, where we looked at trait data from both programs and wanted to see how the predictions would compare if only one set of data was used to predict the the genotypic set in another breeding programs. We evaluated a number of traits, but I'm showing only 2 traits here. Shatter resistance typically has high heritability. Grain yield, as many of you know, is quite affected by the environment and has lower heritability. So for shatter resistance, regardless of the program, the data, whichever program was used, overall, the predictions were quite good across both programs. Not so much the case for grain yield, quite up and down, and nothing too uniform in terms of prediction. So the takeaway from application of genomic selection in this case study was that, let's say if there were 5 new breeding programs to be established within the US or outside, then one could look at traits like shatter resistance that have high heritability and get a fair amount of confidence in the predictions if they're not able to invest plenty of resources in all traits. Whereas for traits like grain yield, it is better that they evaluate it on their own. So that was an interesting finding when the data sets were combined. I evaluated genomic prediction in our own program as well, and I, in particular, use genotype by environment interaction effect in the models, and I wanted to see if they did anything to the models. And I found out that for certain traits, the predictions went up quite dramatically. Examples here show a spike weight where the predictions went up by 15%, spike length 20%, free threshing or threshability by 23%, and grain yield by about 18%. For some traits, there were no change, like 1,000 kernel weight. But overall, it predicted better than models that did not have G by E interaction effects incorporated, which is shown by the bar with the red dots. I used the same model to estimate genetic gain for the traits I was interested in. Here, they're sorted by the highest value. But let me walk you through the table. On the very left, I have trait and their units, and then trait variance, standard deviation, selection intensity. The R here is prediction ability or accuracy from prediction models, and this is the estimated prediction with the actual trait value. The highest estimated gains were promised for grain yield and 1,000 kernel weight. I guess I'm more excited about 1,000 kernel weight because seed size has been a tougher trait to improve, and I'll show you some data later on. However, how does this compare with what we see in the field? We'll find out starting 2021, because this was the first time I applied the the model on the C4 population, and as I said earlier, C5 population is just now in the field, so we'll harvest the data in summer 2021 from St. Paul and Lamberton. And here's the overall breeding scheme we implement at the university with genomic selection models pretty much at the core of what we do. On the left, we have what we call population improvement, and on the right, we have cultivar development. So let's look at this one at a time. On population improvement, we're going to start at training population again. We evaluate nearly 600 plants at 2 locations for 2 years, although we are trying to bump that to 3 years if we have continued source of funding. We genotype using GBS in-house, and we combine that data to train our genomic selection models. And while that is happening, we genotype 3,000 to 4,000 plants as part of the larger breeding population. And once we have the same set of genotypic data as the training population, it's passed through the best models to predict performance of all these plants. And from that, I select up to about 100 plants, cross them in the greenhouse, and that gives rise to the next training and breeding population, or the next cycle. And this whole process, the population improvement process, takes us 2 years, and that's why we say our breeding cycle takes 2 years. We could push it to 1 year, but given the capacity we have, we're pretty comfortable with 2 years. Now, on the cultivar development side, the best plants we see in the field are cloned and transplanted in what we call synthetic crossing blocks that are physically isolated either by a pollen barrier like winter rye, or just by planting in different field sites so that there's not much pollen contamination across the blocks. And usually it's either 7 or 10 genotypes that are replicated and cloned. The seed harvested from each block go on to yield trials at multiple locations in the state. And the best 2 to 3 that come out of the yield trials go to a seed increase, while at the same time, the yield trials are still going on. And based on all the combined data, we toss out, well, we keep one and release the best as the variety. So MN-Clearwater, which I'll talk about briefly in a second, went through all this process minus the genomic selection models because we were still training the models at that time. But any other future variety that will come out of our program will have gone through this exact scheme that you're seeing on this slide. We have been able to improve traits using genomic selection, and these are some of the results after 4 breeding cycles. On the left, I have seed shatter, where we don't have data for cycle 1, but from cycle 2 to 3 to 4 on average, about 6 to 8% improvement. For free threshing, which means what percent of the seeds are dehulled or naked coming out of the combine or lab thresher, that is much faster rate, about 15%. But please do note that I'm only using data from the portion that I select on. That's why these numbers are a little higher. But on population scale, the numbers are single digits, but nonetheless they are on the upward swing. And on the right, I have seed size, where we are making slower progress. These are the largest seeds, not percentage values. So in percentage, it would probably be 2 to 4%, so much less than the other traits, but still on an upward trend, so that's promising. And here's some examples in terms of what seed size we see currently in intermediate wheatgrass in our program. Column 1 here is Rush, a forage variety, so some of the smallest seeds. 2 and 3 are MN-Clearwater from 2 different environments, not too different. Columns 4 through 7 are some of the largest seeds we saw in our cycle 4 population. And column 8 is Lang MN, the hard red spring wheat. And obviously, there's a stark difference between wheatgrass and wheat. And to elucidate that point further, here's a scale from 1 to 40 grams. The UMN wheat variety candidates, the 1,000 kernel weight or the seed mass ranges 30 to 37 grams. Note that despite looking big and plump, there is still quite a considerable amount of variation in wheat because there is genetics and environment constantly affecting the trait. Though compared with intermediate wheatgrass variety candidates, which is right about here, 5.6 to 8.5, we are on the opposite end of the spectrum. So we have a lot of catching up to do. This bar, the underlined bar, are some of the largest seeds we saw in our cycle 4 population, as I mentioned down here. But this gives me hope. We are inching towards wheat slowly. The length, you will notice, is actually longer than that of wheat, just that the width is smaller, and that's a trait, or that's a property we would like to improve. Another observation here is that the wheatgrass seeds have a varying degree of kernel color, and that's pretty typical, at least in Minnesota. We have not yet looked at genetics behind it, but we have an undergrad student who is working on a UROP to do some association study to identify what loci in the genome are controlling kernel color in wheatgrass. Continuing with the breeding program, here are our breeding goals. Of course, we care about the grain yield. On our long list of things to improve, we have now yield longevity, which is in Minnesota, the wheatgrass yields tend to be on the decline after second year of harvest or by stand age. Being a perennial, that's not ideal. We want a farmer to have good productivity in year 3, 4, and so on. So that's a trait we're actually working on in collaboration with the Land Institute and also our agronomists here on campus. Want larger seed size, as I mentioned. We do not want the plants to shatter in the field from rain, wind, or mechanical threshing. We want dehulled or naked seeds readily coming out of the combine or a lab thresher. Want good spike traits. We want optimal height, not short height. And I'll get into that next slide, why we do not want short height. Resistance to lodging, resistance to diseases, obviously, with Fusarium head blight and ergot being the two main ones for us in Minnesota. We also give our materials to food science faculty, Dr. Ismail and Dr. Anor, to evaluate the use of wheatgrass flour and seed in different food and beverage products. The picture on the top right here is the crossing block I mentioned earlier. As you can see, there's winter rye kind of surrounding this block from another one that's right here. It's barely visible. And the bottom picture is our space plant selection nursery, also our training population. This is where the most diverse plants can be found. A quick look at how the traits correlate in wheatgrass. This is cycle 4 data, and there's a lot of numbers, a lot of traits, but as I mentioned, I'd like to sort of guide you through the table by focusing on first height. Let's jump to this number right here. It's a pretty strong correlation between height and grain yield. The taller the plants, more the biomass, I believe, and it just leads to more yield. The smaller, the shorter the plant, which in my hypothesis are inbreds, do not produce much. That is why taller plants tend to produce more grain and also have larger seeds, as shown by the correlation here with 1,000 kernel weight. So based on that, a dwarf wheatgrass or a short wheatgrass as of now is probably not a possibility, but that's not to say that 5 or 8 years in the future we won't have a short wheatgrass that would produce the same as the tall wheatgrasses we have now. Another trait to look at is free threshing or threshability. Almost no correlation or no association with any of the traits except shatter resistance and seed width. And it's been a tough one because I've noticed in my program that the biggest seeds in wheatgrass tend to be hulled, and of course it takes an extra step to dehull them, which we want to avoid. And to compound that problem, anything that shatters nicely does not tend to thresh as well. Of course, there's always a portion of the germplasm, the top, I'd say off the top of my head, 15 to 18%, that have good threshing and good shatter resistance, but overall they have negative correlation. And as expected, there are seed traits that correlate well with 1,000 kernel weight and grain yield. Continuing with the breeding work, let's talk about the release of MN-Clearwater, which was our first Kernza perennial grain cultivar with a few more in the pipeline. This was released based on its performance data from multiple sites in the state of Minnesota. And except in St. Paul, MN-Clearwater, by the way, is this pink bar, or the pink colored bar. Except in St. Paul, it performed pretty well in all other sites. And all this data were collected with assistance from Dr. Jake Youngers and Dr. Craig Schaefer. So special thanks to them for all the data. On average, MN-Clearwater produces 700 kilograms per hectare, or 621 pounds per acre, which is not much compared to wheat or barley or other mainstream cereal crops. It has minimal lodging and negligible disease levels from what we could see in our trials. Another comparison chart here, MN-Clearwater is at the top, sorted by grain yield. I would like to focus your attention to plant height, which is the shortest of the bunch. Lodging was also pretty much non-existent in our trials. However, where it suffers is the grain size. So 1,000 kernel weight was 6 grams compared to other candidates in the cohort that are larger seed, especially this MN-1501. Pretty large seed, but again, going back to my point of not threshing well when you have large seeds, the threshing was pretty bad. So we discontinued this one early on. But overall, it was a very pleasing candidate out of the 5, and among the contemporary varieties, it did better. So we went ahead with its release. The last slide on MN-Clearwater, this is what the seed looks like on the left here, coming out of the combine. And after dehulling, that's how the seeds are, pretty small. Here's the cross section. You compare that with harder spring wheat and soft white winter wheat. It resembles fairly similar to what winter wheat, soft wheat has here. And as a matter of fact, all food science research has shown that all known wheatgrass successions they have soft endosperm. And that was why we got into in this project where we wanted to identify any wheatgrass genotype that had hard endosperm, if any. And the rationale behind this work is that if we can find a genotype that has hard endosperm, that probably means a different protein profile, and that possibly means a variability in terms of the functionality of the flower. And that would immediately, if we're successful, expand the portfolio of current food and beverage products that can be made using wheatgrass flour. And this is a project we are carrying out in collaboration with, again, food science faculty, and Kevin Dorn here helped us in the beginning with sequence alignment. First of all, a brief background on the hardness genes and why wheat endosperm is hard or soft. There's 2 genes making the pure indolin protein in wheat, both on short arm of 5D. When in wild state, you get a soft wheat, and any mutation in either of the alleles leads to a hard wheat. So given that we had a genome sequence, we wanted to know, is there any similarity, or can you even find anything similar to what wheat has? And lucky enough, we found both PIN-A and PIN-B homologs with pretty much full sequence alignment, yet there's a lot of sequence diversity. As you can see, the blue color, the blue shade shows diversity in terms of the alignment. Once we had the sequences identified, I designed PCR primers on those sequences. I screened 24 diverse wheatgrass genotypes and did Sanger sequencing on those. The sequence alignment showed almost no variation in the PIN-A gene for wheatgrass, whereas for PIN-B, I found 4 fairly uniform mutations across the genotypes here. Then Cas markers were designed to target all mutations. And after that, I screened about 900 genotypes from 14 families of the cycle 3 population, from which I identified 65 that were either homozygous or heterozygous for those 4 mutations. I physically separated those plants in 4 groups and put them in different greenhouses for crossing among each other in an attempt to get homozygous loci for any of the mutations. Then the harvested grain, again, from the crossing of 65, well, of the 14 families were given to Dr. Ismail's group for single kernel hardness assay. And here are the results. First of all, let's look at hard red wheat right here in the white shade. The numbers, one is close to 5,000, but very well above 4,000. And on average, none of the wheatgrass families that we identified from marker screening and crossing come close. However, these are much larger values compared to the bulk wheatgrass samples from the years 15, 16, and 17. So already this was quite promising that maybe we do have something coming from the mutations. Then if you look at single seed numbers, these four stood out from two families, the 255 and 333. They came really close to what wheat had. So we're pretty excited. We ditched all the others and kept these. Well, we're given the seed back for all, but I only selected these plus some of the other individual seeds that had harder profiles. I planted, some survived, but as seedlings, but they all died. None but two made it to adult phase, and those were these 255-9 and 333-4. So in a way, we got lucky because two of the hardest seeds survived. Maybe it has something to do with embryo survival. I'm not sure, but those two plants, once they were a bit bushy, I cloned and I self them as well, despite of inbreeding depression, and I also crossed them in the greenhouse. And you can, and the seeds were handed back to Dr. Ismail, and you can see the results again. The numbers are still, they match what we saw overall here for the ones that came after marker screening. Yet this time around, the 333-4 had better numbers compared to 255. Another thing to note is on selfing of 3334, or when it was used as the mother plant, the numbers were very similar. So it's just one plant, one sample, can't make any conclusion, but it's possible that maybe this particular trait is maternally inherited, but again, not definitive until we find more genotypes that show similar profiles to this crop. So the seeds also were returned from this lot, which I either germinated or just got DNA by crushing the seed. I resequenced to confirm the mutations as we saw before, and they're all there, except there's a couple weird samples. So the summary so far on this project is that we may have identified one genotype, two possibly, but one that's more promising with a semi-hard endosperm. I'm calling semi-hard because the numbers are well below what the hard wheat shows, but these are way better than what the regular bulk wheatgrass samples have to offer. The next steps in this project are STS page to look at the protein profile, and we've already given some seed that were obtained from selfing of the 333-4 in the field to Dr. Ismail. And I hope that this summer we can get more seeds so that we can look at the effect of milling on flower particle size and starch content, which will be done by Dr. George Anor. The last project I want to talk about is diseases in wheatgrass. So overall, diseases don't affect wheatgrass production as much, although it doesn't mean that it's safe from diseases. There's four main ones we see quite regularly in Minnesota. Panel A here is Fusarium head blight, caused by the fungus Fusarium graminearum. B is bacterial leaf streak, caused by bacteria Xanthomonas translucens. And C is ergot. D is spot blotch. And in my experience, B and D, bacterial leaf streak and spot blotch, do not really affect the yield, but A and C, Fusarium or ergot, when present, they can do some damage. So these are something we actively select against in our breeding program. And recently we've obtained new funding to carry out genetic mapping for all these diseases. And that work we're doing in collaboration with the Land Institute in the years 21 through 2024. But we did screen our cycle tree between 2017 and 2018 just to see how our training population fares against this disease. The method was grain spawn, and Dr. Ruth Dilmacki in Department of Plant Path helped us with that. We had a misted nursery in St. Paul, and I had about 600 some genotypes. And as you can see from both charts, there's quite dramatic decline in the disease incidence and the coefficient of infection. The plot on the left is a spike count and the frequency of infected spikes, and the right is coefficient of infection. But overall, of the 600 some plants, there were 38% with absolutely no diseases, and of the next 35% that had only one or two spikes infected, the average incidence was less than 30%. So by and large, quite resistant on a population level. We did carry, well, before I move on, because of these results, we discontinued Fusarium head blight screening. I still take data, a binary presence absence data, to deselect plants that show high levels or even any alarming levels of Fusarium, but we don't do any active screening anymore. But if the disease becomes a problem, we can obviously switch back and continue it. So we did map for resistance to Fusarium head blight, and we found 16 QTL on 18 chromosomes, shown by the square blocks. The circles are for bacterial leaf streak, which I'm not describing today. And these 16 QTL I used as fixed effects in genomic prediction models to see if there was any improvement, and there was up to about 11% improvement in prediction. So not a whole lot, but still something I could implement in the program, given that I still take the binary 0 and 1 presence absence data for Fusarium head blight. This is my last slide. I talked the whole talk was about wheatgrass. Now we're looking at soybean. This is the progress the soybean community has made in terms of improving its yield, and this is just one trait. I'm sure they've improved a lot of other traits, but in many levels, the lessons are similar when compared to wheatgrass. So in the early 1920s, the yield level of soybean was about, I believe, slightly over 700 kilograms per hectare, the current level of wheatgrass. In 100 years, they've pretty much improved that by 5-folds, so roughly doubling it every 2 decades, more or less. There was now soybean by the time it came to the U.S. was already domesticated. It was not in the wild shape that wheatgrass was when work began back in the 2000s. But nonetheless, soybean did not enjoy the genomic tools or genetic resources that wheatgrass has now. The reason I think soybean enjoyed this progress is because there was interest from the farmers, interest from public, private sectors, a lot of research, resources being poured in to make soybean the crop that we see out here today. Wheatgrass can, I think, enjoy similar levels of success, given that there is this continued support from public, private sectors and interest from growers, and all of those are currently present. As you heard in the very beginning when Dr. Anderson spoke about the amount of funding that we've obtained in the past few years, the resources that the community has developed, the enthusiasm of growers, where everyone is asking when and how I can grow Kernza, interest from local and national business partners who want to make food or beer. So everything is there. I just think the support has to stay in place and continue. And maybe when we come back in 10, 15 years, we can track where we're at. But I'm pretty confident, based on the work we're doing, based on the work the Land Institute and other collaborators are doing, we can go on this trend. So with that, I'd like to end on this slide, thanking a lot of people that have contributed to what I presented today. Special thanks to Dr. Lidihan at the Land Institute, Dr. Jesse Poland at Kansas State University, Dr. Steve Larson at UST ARS. They've been solid partners in furthering wheatgrass research. Our source of funding, of course, very valuable, comes mostly from the Forever Green Initiative through the Minnesota Department of Ag. General Mills supported us, still is supporting us. The recently acquired FFAR. Kernza Cap is not here, but that is going to be quite vital going forward as well. and all the other faculty, grad students, postdocs, undergrads, technicians, the research and outreach centers that have helped to bring Kernza this far. So thank you all for your attention, and I think we can move on to questions.
All right, thank you, Prabin. All right, the first question came in early during the seminar. So everyone can continue to put questions in the Q&A, or if you raise your hand, we can open up your mic to ask it live. So the first question is, you stated population improvement was a 2-year cycle. What about cultivar development side of things?
Yeah, sorry, I skipped over that. Let me see if I can go there. Off the top of my head, I'd say it takes us about probably 5 to 7 years. Crossing block, well, going from here to the crossing blocks is 1 year. Yield trials, at least 2 years. We're thinking about putting that for 3 years, again, for the yield longevity issues. Seed increase, we have to have 2 years to get enough seed to grow, or to have enough to pass it on to the growers. So all combined, a minimum of 6. So anywhere 6 to 7 years, I'd say.
Okay. Yeah, so those synthetic generations, it's if we do a maximum seed increase, which is, you know, consecutive intercross generations, we go from SYN 0 is that starting crossing block. SYN 1 is the seed harvested from that gets intercross, makes SYN 2. So we're trying to have a situation where the growers would get mostly SYN 4 seed, but the extra time is for whatever additional testing we want to do. So if we just maximize our speed, we know we've got something really good. We could do it in as little as 4 years, but in reality, it's probably going to be that 6 to 7 year time period. Next question, is harvest timing consistent between selections? And I think this may be getting at maybe how you're evaluating shattering, I would guess.
Yeah, no, that's a great question. As breeders, we don't really, we time our harvest at physiological maturity, have to work around the weather conditions. We have not done any trials on harvest timing, but Dr. Jake Youngers, they have, their group has done some work, and if not published, I believe the work is going to be published soon, where they've looked at different levels of moisture content based on harvest timing, when is the optimal harvest time, and how that impacts shattering and yield. I've seen the data shown in our Forever Green meetings, and I'd like to think it's published, but we have not, but we rely on the information that our agronomy team provides us to sort of time our harvests as well.
Okay, I have 2 questions from Peter Innes at University of Colorado Boulder. The first one is high heterozygosity and seed heterogeneity at odds with creating a uniform and reliable crop for farmers?
Oh boy, I think that definitely impacts how the plants look. If you go out in the field, some are short, some are tall. Nothing too crazy, probably because of that sort of pollen sharing among the plants. But there is quite a lot of diversity in terms of height and other characteristics, even within a cultivar. That's for sure.
Yeah, I would describe it as almost hopeless, but it's sort of an acceptable thing with the cross-pollinated species. You're just not going to have the uniformity in a field like you would with a self-pollinated crop. His second question is, where and when were the original collections for the breeding program made? Do you think you have a full representation of wild population genetic diversity? Are collections still being made?
In terms of genetic diversity, let's work backwards in that question. I know that we are sort of, because each program selects for certain traits, so we are moving away from what the Land Institute has or had back in the days. I'm not sure if they still maintain that population, but if we were to compare, the population would look quite different. Are collections being made? I'm not sure. But as far as I know, the starting population that was at the Rodale Institute used nearly all accessions that were present with the USDA core collection. To my knowledge, not much collections are being made, as I don't really think a whole lot of diversity exists out there. Plus, if we were to start, it would be going backwards in terms of, sort of unwinding the progress we've already made. So that's another challenge there.
Yeah, and I would add that we observe a lot of phenotypic diversity even within our relatively narrow germplasm base. So we think we can make a lot of genetic progress in what we're currently working with. Next question, is time to physiological maturity consistent among lines?
For the most part, yes, but not always. So some say on a single space plant nursery, some plants will have undergone a bit more shattering than the others by the time we're ready to harvest all. Within a variety large plot, there's shattering. That could be because of non-uniform maturity, but there's always some losses and some gains when we're working with a crop like this.
Okay, and let's see. Interesting presentation, Dr. Bajgain. Can you please explain why you need 6 to 8 individuals per family in the training population? Heterozygosity, heterogeneity, are there other crops like maize panel?
We mainly do that to have, well, one, to have the pedigrees represented adequately so that we don't lose that diversity. The second is that even among a family, we see a lot of variation in terms of trait phenotypes. So we want to include as many as possible without burdening the breeding program to make sure that we are sampling that diversity to give us that boost in genomic prediction, while also allowing us to select the best plants in the field. And note, unlike maize, I do not think we have that panel in wheatgrass yet. We do have some nested association mapping populations, but not a panel as such.
Okay, and I'm seeing one more question. How was threshability evaluated?
It's the percentage of naked seeds or dehulled seeds that coming out of the combine. It's typically my two eyes. We are thinking about using a scanner to sort of making the process more uniform, but that will be time consuming, I'm imagining. But so far, it's just my visual inspection.
Okay, I had a couple of questions that came to mind as well, so I'll go ahead and ask them. So you said that the synthetic crossing block parents were selected based on phenotype only, or they were selected based on phenotype, but do you consider the predictions as well as the phenotype, or phenotype alone?
For the synthetic crossing blocks, it's a true phenotype we see in the field, because then I know that the plants in the crossing blocks will continue to maintain the performance they had in the training population or the space plant nursery. Before making the crossing blocks in the future, I do want to make simulator crosses in order to see which ones should be combined. But again, as for traits, it's 100% based on the values seen in the field.
Okay, and the training population, so you're every 2 years, it's a new cycle, a new training population. Are you planning on using previous training population data at some point and just building on the existing data, or what are the barriers to doing that?
There actually are no barriers. That is an upcoming study. I was just speaking with the previous postdoc, the previous Kernza breeder over the weekend, and we're coming up with a plan to evaluate maybe same number of individuals per cycle so that the population sizes stay the same. But that's an upcoming study. Hopefully I'll start it by mid to late summer.
Okay, and then another question came in. What's your methodology for testing, measuring shattering? Is it similar to threshability?
That's also manual. I run my fingers along the spike and see what percentage of spikelets drop. Some people do the spike dropping method. That I found is it gives data taken with that method, at least the ones that I've seen being implemented, the way it was implemented, it leans towards shatter resistance. And I've been more comfortable doing it the way I was trained in the beginning, but we have a new grad student who will be looking at some of these traits in a set of parents for all the cycles, and she will be looking at protocol development for certain traits. So hopefully we can come up with a better, more uniform, more reliable method for that trait as well.
Okay, thank you. I don't see any more questions or hands raised. At the beginning, I neglected to welcome prospective graduate students. So this is officially the first event in our 2021 graduate recruitment activities, which continue on through the week. Most activities are Thursday and Friday. So this is a good showcase of one of our former students and what he's working on today with a perennial grain crop.
And just to remind all the students that are looking at the program, this is also the seminar series in which you will present your research proposal. So this would be a very good example of the type of presentation you would have the opportunity to give to your peers and faculty and other community leaders as you present your research program, the type of feedback that you can get during that presentation. So welcome to all of you that are visiting this week. Looking forward to talk to you. For me, I'm Don Wise, talking to you at the Forever Green lab meeting on Friday.
All right, Cody, I didn't miss any hands raised, did I?
Nope, I think that's it.
All right. Thank you, Prabin. Thank you, everyone, for attending.
Thanks, everyone.
Good job, Prabin.
Bye-bye. Stay warm.